My name is Xinyi. I am a postdoctoral research associate at the University of Illinois-Urbana-Champaign in the Department of Natural Resources and Environmental Sciences. My advisor, Dr. Carmen Organte, and I are working on a multi-site evaluation of microbial community composition in the NRCS Dynamic Soil Properties for Soil Health Initiative. Today, we are presenting some preliminary results on evaluating soil health using biologically based indicators. We are interested in investigating soil health because of its critical role in our daily life through supporting key ecological functions. Soil health indicators must be able to distinguish changes in management. Dynamic soil properties can serve as a tool to evaluate and monitor soil health because these are properties that can be changed through the land use change and management. Soil microbes are associated with many aspects of soil health, which are not limited to nutrient availability and carbon sequestration. The soil microbial component is also a sensitive indicator that distinguishes between different environment and management. Different soil microbial groups depict various microbial physiological traits. Characterization of the soil microbial community composition can be done in multiple ways that using different methods. One example of this is through the identification of specific types of fatty acid in the phospholipid membrane that characterize different microbial groups. These fatty acid methods estimate the microbial biomass and abundance of microbial groups. Phospholipid fatty acid profiling, also known as PLFA, is one of the measures that is being evaluated in the DSP4 Soil Health project. In recent years, PLFA has also been available commercially for soil health assessment. Managing soil health encourages the use of management practices that improve soil function. Thus, in this project, we are evaluating soil health management against business as usual practices and their influence on microbial communities. We are also identifying soil properties and environmental factors that can help us better understand the variabilities introduced by the microbial community dataset. We included 5 study locations in this study, including Washington, Texas, Kansas, Minnesota, and North Carolina. The soil samples were collected between 2018 and 2019. For this study, we are using soil samples from 0 to 5 and 5 to 10 centimeters. In total, we have 224 soil samples. In this slide, we have a summary of the management practices at each of our study locations. Something that comes across is the BAU is represented by annual cropping with conventional tillage and mulch tillage, while soil health management includes fields that has been less physical disturbance, such as no-till or perennial hay. We used soil data from the DSP4 Soil Health project specifically for the work that we are going to present today. We are using the PLFA data. We are also using soil and environmental data such as clay, soil organic carbon, aggregate stability, pH, and epoxy. In addition, we collected long-term normalized difference vegetation index (NDVI) data through MODIS database by using Google Earth Engine, and we used it as an explanatory environmental indicator. NDVI reflects the vegetative cover while also correlates precipitation and temperature data. The analysis of enzyme activities is also undergoing. We are not going to present it on today. We used a variety of multivariate statistical methods, including principal coordinate analysis, distance-based redundancy analysis, DBRDA, and permutational multivariate analysis of variance to sequentially address our research questions. These research questions include how microbial community varied across 5 study locations, how explanatory soil properties and environmental indicators explained variations in the microbial communities, and the influence of location, management, and their interaction on microbial community. Let's start the results section with these bar plots showing the abundance of different microbial groups across different states and systems. The red color is business as usual, and the blue color is soil health management, SHM. Soil health management leads to greater abundance in the microbial groups of gram-negative and gram-positive bacteria. You can see in the middle of the figure. Except the cancer state. Gram-positive and gram-negative bacteria are common in rhizosphere, where gram-negative is more closely tied to routine zones than gram-positive bacteria. We also found similar pattern in the AMF, the left corner. This is that soil health management leads to greater abundance than BAU. In all 5 states, the soil health management leads to greater abundance in the actinomycetes, which is a gram-positive bacteria active in decomposition of soil organic matter. In general, soil health management improved abundance in most of the microbial groups. We further explored the variation across 5 study locations and 2 systems. The figure shows the results from the Principal Coordinate Analysis, PCOA. The first 2 axes of PCOA explained roughly 35% and 6% variations for PFA. The closer or overlapped groups means they have similar microbial community composition. We can see there is a clear separation of Minnesota and North Carolina from other states. Texas was partially overlapping with Kansas and Washington, while partially standing out from the clusters. In North Carolina, the 2 systems, BAU and SHM, have distinct microbial community composition. The soil samples from different states were collected at different times of the year. The Kansas and Washington states are both sampled in October 2018. That might be another cause of these 2 states are overlapping a little. We used distance-based redundancy analysis to better understand how different explanatory variables explain the variation in PLFA. The dominant driver is SOC, which explained about 34% of the variation. Following soil and carbon, and DVI explained 50% of the variation. These 2 adding up explained almost 50% of the variation in PFA. Carbon in the literature has a positive relationship with PFA, as SOC is the main source of energy for soil microorganisms. Soil pH was reviewed as critical indicator of PFA because some of the microbial groups were sensitive to the acid conditions. In this specific case, it only explained 3% of the variation. Overall, PFA has a high sensitivities to these explanatory soil properties and environmental indicator. We investigated the influence of location, management, and their interaction on microbial community composition. Here are the results from PermaNova, which is analogous to ANOVA test. Here are the results from 0 to 5 and 5 to 10, and the combined 0 to 10 centimeters. At the 0 to 5 centimeters, we found the location, management, and their interactions were all significant on microbial communities. However, at the 5 to 10 centimeters, only location was significant. There was no influence from management and no significant influence from the interactions. In summary, we found location is the dominant driver of microbial community, while management plays a role on surface soils. Based on these results, we can suggest that soil health management and evaluation needs to be region-specific. Summarizing our presentation today, we investigated microbial community across 5 states, our study locations, and 2 systems to answer the questions of how microbial groups variate across the state and what are the drivers of the variations. We found location is very influential on microbial community at all depths, and so organic carbon and NDVR are the main drivers of variations. We are still working on the enzyme activities. We would like to acknowledge the funding from NRCS, and we appreciate the efforts from our collaborators in this work. Thank you.